Meiosis is a form of nuclear division that produces 4 haploid nuclei from one diploid nucleus. Meiosis involves 2 cycles of division, meiosis I and meiosis II, which result in 4 daughter haploid cells. Prior to meiosis, during interphase, the DNA is replicated so that all chromosomes consist of 2 sister chromatids. Then begins meiosis I, which allows reduction division, meaning that cells begin with 2 copies of each chromosome and end with only 1. The first stage of meiosis is prophase I, in which chromosomes become visible due to supercoiling. The replicated chromosomes form closely linked homologous pairs, which have 2 chromosomes and 4 total chromatids. At this stage, non-sister chromatids may cross over and exchange equivalent segments of DNA, which we'll revisit later in the video. During prophase I, centrioles migrate to opposite poles of the cell and spindle fibers start to form. The nucleolus and nuclear membrane also disintegrate. In metaphase I, the spindle fibers attach to the centromeres of each chromosome and gently pull to align them along the equatorial plate. Maternal and paternal homologues show random orientation towards the poles. During anaphase I, the spindle microtubules shorten, which pulls the homologous chromosomes apart towards opposite poles. The sister chromatids remain connected at the centromere and move to the same pole. As the chromosomes arrive at the poles, the first meiotic division ends. Each chromosome still consists of a pair of chromatids. These chromatids partially uncoil and a nuclear membrane then reforms around each nucleus. Cytokinesis usually also occurs during telophase I, which results in 2 daughter cells with haploid nuclei. Then begins meiosis II, in which the chromatids are separated into haploid cells. During prophase II, chromosomes condense again, the centrioles migrate to the opposite poles, and spindle fibers start to form. The nucleolus and nuclear membrane disintegrate. During metaphase II, the spindle fibers attach to the centromere and pull the sister chromatids to the equator. In anaphase II, the centromeres divide and the chromatids are moved to opposite poles by the spindle fibers. Once the sister chromatids are separated, they are then referred to as chromosomes. When the chromosomes reach opposite poles, they uncoil. This is then followed by the nuclear envelope formation and cytokinesis. Meiosis is now complete, resulting in 4 haploid daughter cells. All 4 of the daughter cells are genetically distinct. This is due to the crossing over that occurred in prophase I. In prophase I, the homologous chromosomes pair up and form a tetrad. In the tetrad, non-sister chromatids have the same genes but may have different alleles. Crossing over occurs when equivalent portions of the non-sister chromatids are exchanged between the homologous chromosomes. The point at which the crossing over occurs is called a chiasma. The crossing over creates new combinations of alleles that were not present in either of the original chromosomes. After the crossing over has taken place, the tetrads complete the process of condensation and begin the metaphase I. This is the reason for the genetic diversity among the gametes produced by the parents. Meiosis has allowed 2 diploid individuals to each contribute half of their DNA to produce offspring in sexual reproduction. The fusion of these haploid sex cells then produces a diploid nucleus, thus allowing a sexual life cycle. Thank you for listening.